Hello everyone, welcome to the video, and today we will be starting a new series of videos on the topic called molecular processes of the cell. So this is a kind of the format that I'm trying here, where you can also see me at the same time. I have already written some of the basic concepts regarding the topic, and then we'll pick up one chapter at a time. I'll try to make as many as videos on that particular topic, and then we'll just move from one topic to another one, understanding what are the important concepts in there, discussing what are the important questions that can come in different exams. So we'll try to dig into the detailed mechanism of that particular topic as well. So in this particular video lecture series, I'm starting with the molecular processes of the cell. Here you can see molecular process of the cell title is written, and let me show you. I have quite significant amount of material that is already there, and then at the same time I'm going to add more into it. So one by one, what we will do, basically we'll choose the topic, we will discuss that topic in detail, and then we'll move on to another topic which is going to be connected to the previous one. So I recommend you guys to follow through, and also if you can read about that particular topic, it will be great. It's going to help you, but you can just watch the video. It will definitely help you understand that particular topic. In this one, we'll try to cover the molecular processes, including DNA replication, transcription, translation, and so on. There are so many things that we need to understand in molecular processes. So I'm sure that this is going to strengthen your biochemistry as well as molecular biology subjects, and I expect that, okay, you're gonna like all those videos. It will be a little bit informal kind of video where I'll try to also draw. So sometimes you're gonna have to be patient, and at the same time also just watch the full video so that you can understand what are the important things in that particular topic. Okay, so as you can see, the layout will be quite simple. We'll have on the left side, where I'll just try to draw right few things. On the right side, you can see me and I can explain those particular things. So I hope that this is going to be a little bit engaging, and then you will be able to understand different topics as well. Okay, so this one will be more introductory video, and then we'll jump onto the specific topic, and then slowly we'll dig deep into that particular topic. So as you can see, let's start the topic, and here you can see the title that I've written is molecular processes of the cell. So here we have molecular word that I'm using. Okay, let me use the dotted line. I, for some reason, like the dotted line, and then the straight line. I don't know, maybe it gives me the freedom where if the line is not straight, it definitely helps me out in that case. So you have few important things: molecular, and then molecular processes. That includes various different kinds of processes. We'll try to cover as much as we can. We'll start with, as I told you, we'll start with gene, DNA replication, chromosomes, and so on and so forth. And after that, we'll move on to the other processes as well as, and try to discuss those processes as well. And next, I will also start a new series of lectures in another dimension of the biological sciences, and in the same way. So I hope that is going to be helpful for you. I'm being a teacher, I'm being a professor in the area of the biochemistry, cell biology, and I'm teaching microbiology, biotechnology subjects. So I hope that my experience with the students is definitely going to help you also to understand particular topic. So we'll start with that, right? Molecular processes. I hope that this is clear. And cell, what is cell? We all know we are talking about the eukaryotic cell specifically, but we can also go into the because it's simplicity. So we'll start with the bacterial cell, which is the prokaryotic cell, and we all know, if you are not aware of prokaryotic cells, then please do watch my videos. I made a lot of different videos on my molecular biology of bacterial cell, structure of bacterial cell, so that you understand what is bacterial cell and how it looks like. Okay, so this is I have designed here. So we'll try to cover as much as we can in both cases, but we'll see, right? Coming on to the next one, which is here, which I have written is the biochemical, and then, as I told you, this is just the foundation that I made. In addition, I will keep on writing new things so that it's all. A lot of things are missing in this one, so that is why I've just made out the layout, and then we'll just fill it up. So we'll be studying biochemical mechanisms or pathways, you can say, biochemical mechanisms, to understand what are the different molecular processes and how they occur inside the cell. Next is the molecular structure of various biomolecules. So let me write it down. Molecular structure of various biomolecules that are there, how they function, what is the structure, and then next is how genetic information. So this is again how genetic information is basically transferred. So I think we should basically use the word transmitted in this case, right? So I'll just write it down. Transmitted from one generation to another generation. One generation to another generation in high fidelity, right? So what that means is with high accuracy, and we know that genetic information that we have in our DNA needs to be transferred in high accuracy to the next generation. So that's the another thing we'll be trying to understand. Okay, so next is, let's move a little bit down, and next is the discovery of the DNA molecule. The discovery of the DNA molecule is one of the various big discoveries that has been done in the area of life sciences, and discovery of the DNA is one of the major, I would say, stepping stone when it comes to the advancement of the biotechnology. And when it was done, so discovery of DNA, right? These are the two people, James Watson, Francis Crick, and what was the date, right? 1950. So you have 53 here. 1953 is the date when the structure was discovered. And what we have done in addition to that is basically discovery of the DNA molecule, and what was the structure. So it was double helical structure, right? So it's a double helical structure of the DNA molecule. So these are the important things that we need to remember, that who are the people discovered the DNA structure. James Watson, Francis Crick in 1953, and they have solved the structure of DNA molecule, and they found out that it is double helical in nature. Double helical means you have one and then you have second structure, second helix of the DNA molecule, and we know that you have like different base pairs. They are interacting, they're forming hydrogen bonds with each other, and this is how the DNA molecules will look like. Okay, so this is also clear. Moving on to the next one. So you can see here I have one arrow and then the second one. So what I'm trying to mention in this case is basically how the structure was solved. So here I think let's see, solve the DNA structure. So what are the tools that were applied? So in this case, X-ray, right? So many of you, those who already know, student of biotechnology, biology, they already know X-ray diffraction was used to solve the structure, and basically solving a biological structure or biomolecular structure, we applied, or they applied, physics and chemistry all together. And you can say, using the help of physical sciences as well as the chemical sciences, the researchers, they were able to find out the structure of DNA molecule. So it is very, very important, especially when it comes to the biological sciences, that the research should be multidisciplinary, involving other sciences such as physics and chemistry to solve the problem in the biological sciences. And nowadays, a lot of different kind of other sciences like computer sciences also jumping into solving the biological and chemistry related, biochemistry related problems, and they have done a great job in that particular topic. Next is DNA structure. So here you can see this is the DNA structure I have tried to design, and you can see this is the double helical structure. You have one strand here, so this one, I'll just write it down, 1 and 2nd, and it's running through like this, right? And they are kind of tangled together and then forming this unique structure, very, very beautiful structure. And we can also download the crystal structure of DNA molecule and try to see how the 3D structure of the DNA molecule look like. So what it indicates also, that just this structure will tell you how the information can be transmitted, because one strand is have the copy of the second strand. So that means one strand, if you have A, T, G, C, these are the nitrogenous bases, those who are aware of the structure, and we'll also discuss that in detail. Anyways, let me explain it here. So for example, you have these 4 nitrogenous bases, and then you will have the hydrogen bonding. A will always form the bond with T. T will form the bond with A, and then triple bond, and the double hydrogen bond will be in between G and C, right? So it will run like this. Let me show you the sequences. If you read the sequence from here, it's A, T, G, C. If you read the sequence from here, so it will be complementary to this one. So you can basically understand what will be the sequence of the second strand if you already know the sequence of the first strand. So the information that is been in the first strand, you can identify what is there in the second strand. So that means the structure itself explains a lot, that the information from one DNA molecule can be easily transferred to the next molecule, or it can be easily transferred from one generation to another one. So it explains a lot how the information is copied and from one molecule to the other molecule. So I told you, like, this is how they are interacting. So this is one strand of the DNA molecule, and this is another strand here, and this is how they are interacting. So it's easy to identify the sequence of the second one. So copying, and if you want to make another strand from these 2 strands, you know that the sequence is going to be the way that I have explained earlier. Okay, so moving on to the next, after the DNA structure was identified, so the scientists used X-ray diffraction. I don't know why I just said X-ray. I think no, it's not X. It's X-ray. It's X-ray diffraction technique that was used, and the data that was generated was then studied carefully, and then from there we found out that this is the structure. This should ideally be the structure according to the data that was generated by Rosalind Franklin. Anyways, we'll discuss all those things in detail, but right now I'm just trying to explain you the basic concepts. Now moving on to the next one is basically from understanding the DNA, we moved on to RNA, we moved on to protein. We found out that the information is getting transferred from DNA to RNA to proteins. And you can see if here this is the DNA molecule, so this is the DNA molecule, and then information is moving. I'm just writing RNA, not mRNA in this case, because we need to understand all those things. So you have the single strand mRNA, probably this is the mRNA molecule, and from there you are making a protein molecule. So this is protein molecule that is being synthesized from the information that was there in the DNA molecule. Next is, what are these processes? So from the DNA, if RNA is being synthesized, how that is being synthesized, this is a part of the molecular process. And here the process is also known as transcription. And then from RNA to protein is being synthesized, that process is translation. There is another process going on in here, is the formation of DNA. From the DNA, we'll call it out. And it is also known as the replication process. So this is the replication process where basically DNA is getting constructed from the RNA, from the DNA molecule. And if the DNA is getting constructed from the RNA molecule, so it is going into the reverse direction. This is known as reverse transcription. This is transcription. If it is going into the reverse direction, this is reverse transcription. So anyways, this never happens where protein is getting back into the RNA. So this is not going to happen as far as current knowledge of biochemistry is there. Next is central dogma of molecular biology. What is this? So entirely whole of the concept we just studied, this entire concept is known as central dogma of molecular biology. So I hope now it's clear what is a central dogma of molecular biology. Moving on to chromosomes, then further we'll try to understand chromosome structure. Let me write it down. And probably topology, and then chromosomal mutation, organization, histone, and all those things will be there, how it is being packed, and a lot of other processes we'll try to cover. And after I conclude this video, I have one interesting fact that I'll share with you here. The fact is basically, when it comes to cellular processes, they are expensive. So let me write it down. They are expensive. Expenses? I don't know. I think I've used the word expenses. It's expensive. Cellular processes, they are expensive. When it comes for the utilization, when it comes to the utilization of the energy, and we all know that we are going to use the units like kilojoule per mole for the energy. The energy requirement of the cellular processes is very, very high in some cases. Here, I'll give you the example. In case of peptide bond formation between two amino acids, so just take the example of one amino acid to any two specific amino acid, average the energy consumption requirement of this approximately, let me just reconfirm it, 21 kilojoule per mole. So this is the energy requirement for forming this peptide bond between two specific amino acid. And then when you are synthesizing the same bond between when it comes to the polypeptide molecules, when you have chain of amino acids, huge molecules all together, then you have 125 kilojoule per mole of the energy requirement. And then in addition to this, you have so many enzymes, approximately 200 more than 200 enzymes. They are known to play an important role when it comes to the formation of the peptide bond between two specific amino acids, but you have those polypeptide chains. That is what I'm trying to represent here, right? We'll study that when we'll study the secondary structure of the protein. So now, since it is important because you have two polypeptide molecule, they need to be oriented. Conformation dynamics is important in this case. So a lot of different enzymes, they are involved in forming the bond, and then energy is obviously being used in this case, and that is very, very high when it comes to the specific bonding between these two amino acids, right? So cellular processes, they are expensive. Another thing is they should be error-free. Otherwise, what will happen? If errors are there, then genes basically they become non-functional, they will be mutated, they will become different. And then if the error is significant or error is dangerous, what will happen? Then there is a possibility of incorporating of any kind of genetic disorders, or any other things can also happen. So this is important to understand that these cellular processes, they are highly regulated. So gene regulation, I don't know if I've written gene regulation, so I should write it down here. So gene regulation as well as gene expression, I can say expression, right? Gene expression and regulation of gene expression is highly important when it comes to the understanding of molecular processes. So there are a lot of operons that we'll be trying to cover. Lac operon is there, tryptophan operon. These are some of the basic operons, but this is important to understand how they work, right? All right, so I hope now at least you understand what we will be doing in this particular series of lectures. We'll try to pick up one topic at a time. We'll go through the topic in detail. Right now, I just try to, introduce you to some of the major and key topics of basically molecular processes of the cell. Hopefully that basically helped you to understand what different types of things we will be covering in these videos, and we'll try to understand the topic in more detail, and we'll try to understand if we can relate these processes to specific diseases or specific functions in the cell, because that is also an important part. Right? So I hope this is clear. What we have done in this case is basically we try to understand what are the basic pathways that are there in case of prokaryotic and eukaryotic cells: replication, transcription, translation, reverse transcription, and those are the important processes. We'll discuss in detail what are the proteins that are involved, and let me tell you, these are highly complicated pathways. So I will try to make it more interested. I'll try to cover or I'll try to design some of the structures in advance, so it's not like very a kind of a video where you're seeing me design things, but it will be more like a coherent video where you'll see the concept and try to understand all those things in a nice and in a simple way. So okay, I hope now everything is clear to you all, and we'll try to cover all the topics one by one. And please if you're new to the channel, then please do support the channel and share the link of the channel so that others can also get benefit from these videos. And if you have any questions, then please do post your questions in the comment section. I'll try to give the answers and try to cover some of the topics related to your comments and questions. All right, okay, so I'll meet you in my video where we'll start discussing these topics one by one. Okay, thank you so much, everyone.